Hello and welcome back, biologists, to your next screencast. Today we're going to continue with Chapter 11 and our discussion of single gene inheritance and meiosis. When we left off, we were talking about meiosis, a type of cell division that produces haploid gametes. Remember that haploid means having half the normal number of chromosomes, and gametes are sperm and egg cells. It's important to know that when gametes are produced, no two gametes are identical. They are all genetically unique. This is due to two factors, recombination and independent assortment. This photo shows how meiosis produces unique gametes and as a result, unique offspring. This is Mary Kate Gosselin and her 8 kids. They all star on a television show called Mary Kate Plus 8. Recombination occurs during prophase 1 of meiosis 1. During recombination, maternal and paternal chromosomes will pair up and exchange DNA with one another. The chromatids will overlap, break, exchange DNA, and reunite. This is called a crossing over event, and it results in the shuffling of genes between chromosomes in every homologous pair. It also results in gametes that have chromosomes that each individually contain maternal and paternal DNA. Independent assortment is simply chromosome shuffling. Because we have 46 chromosomes and only 23 of them will make it into a gamete, each gamete will have a unique combination of those 23 chromosomes. The chromosomes and the genes they carry are distributed independently from one another during anaphase 1 of meiosis 1. After meiosis 2, you create a great variety of different gametes. You can see this in the diagram where we have a cell with 4 chromosomes producing gametes with several different combinations of chromosomes. This infographic provides another representation of independent assortment. You can see how genes A, D, and E, found on 3 pairs of homologous chromosomes, can assort independently to produce sperm that have genetically unique combinations of alleles for those genes. Let's go back to our cystic fibrosis case study. In cystic fibrosis, there's a mutation that causes the CFTR protein to be distorted and not function properly. As a result, the flow of ions and water into and out of cells is disrupted, and an abnormally thick mucus builds up outside of cells, especially in organs like the lungs. This infographic shows how cystic fibrosis can form abnormally thick mucus in the lungs. This thick mucus can then trap bacteria and cause infection. The body's immune system response to this infection can make the mucus even thicker. Let's try to understand how cystic fibrosis is inherited. First, we need to understand that people without CF, meaning they have the normal phenotype, can still pass the CF allele to their offspring. We also need to understand that people with CF do not necessarily have to have offspring with CF. The individual in the photo here has cystic fibrosis. The equipment he's wearing is designed to remove the mucus from his lungs. Remember that cystic fibrosis is caused by a mutant allele of the CFTR gene on chromosome 7. The mutant allele is also known as a recessive allele. This means that you need 2 copies of it if it's going to affect your phenotype. You need 1 copy from your mother and 1 copy from your father. Otherwise, it'll be hidden by the normal or dominant allele. Recessive generally means weaker, and dominant generally means stronger. When we work with dominant and recessive alleles, we normally represent the recessive allele with a lowercase letter. We represent the dominant allele with an uppercase letter. In this example, big A represents the dominant allele, and little a represents the recessive allele. If you had 2 copies of the normal CFTR allele, you'd be called homozygous dominant. This could be represented by big A, big A. You would not have cystic fibrosis, and your phenotype would be normal. If you had 2 copies of the mutant CFTR allele, you'd be called homozygous recessive. This could be represented by little a, little a. You would have cystic fibrosis, and your phenotype would be mutant. The Punnett square is a tool in which we can study and even predict inheritance. We use Punnett squares to study a parental generation, called the P generation, and their offspring, called the F1 generation. Parental genotypes will determine the genotypes of offspring. The Punnett square on the slide shows how a big A parent and a little a parent can produce big A, little a offspring. Punnett squares show all the possible gametes that can be created and all the possible ways that gametes can combine to produce genotypes of offspring. Make sure as you work with Punnett squares, you understand that Punnett squares only predict chances, not actual numbers. The photo on this slide shows Reginald C. Punnett, the creator of the Punnett square. This infographic will walk you through how to use a Punnett square to predict the genotypes of offspring. This infographic shows how there are conditions that are dominant, like Huntington disease and polydactyly. Huntington disease is a neurological disease, and polydactyly is having more than 5 fingers or toes on each limb. Punnett squares still work the same, whether the condition is dominant or recessive. This table describes different inherited conditions that are both dominant and recessive. This table shows different common physical features that are caused by both dominant and recessive alleles. The cystic fibrosis story isn't as simple as one gene with 2 alleles. The severity of cystic fibrosis can be influenced by genes on other chromosomes as well. One example is TGF-beta 1, which influences the immune response to infections caused by CF. The photos on this slide show Emily Schaller living with cystic fibrosis and Emily spending some time with her parents. Punnett squares allow us to follow alleles for conditions and diseases like cystic fibrosis through gamete formation and offspring genotype. They also help us predict the likelihood that offspring will be homozygous recessive for these conditions and diseases. The diagram on this slide shows how 3 different genes with different alleles can influence skin color. This infographic shows how we can use a Punnett square to study the inheritance of 2 different genes, each with 2 different alleles. Understanding how modifier genes like TGF-beta 1 contribute to cystic fibrosis will help us develop better therapies for the condition. Because of our better understanding of cystic fibrosis, we've been able to develop drugs like Kalydeco that are effective at restoring function to the malfunctioning CFTR protein channels. Emily Schaller is alive and well today. She is the head of a nonprofit called RockCF, whose goal is to help people live and thrive with cystic fibrosis.